Hello, people. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, some people still coming. Hang out for a bit, talk. Let's see your faces while you're here. See who the hell you are. People. Maraid Lawless. Haven't seen you in a while. Hi, Maraid. Elijah. I don't think I know you, Crossborn. Elijah Crossborn. We haven't had each other, have we? We have? No. Okay. Angelique. Lee Turner. A lot of new people here. Alex McKieran. I had you last semester, right? Alex? This is yes. This is no.
Yes.
Okay. Stephen McCain. My man, he's here. Tyler Montano. We should talk after class, Tyler.
Okay.
Jordan Fine. Jordan Fine. I know your name. I can't remember your face. Bring your face up. Jordan. Jordan. Evans Cujo. Victoria Chirono. It's not Italian by any chance, is it?
Yeah, it is.
I thought so. I'm a cannoli eater from way back. I love Italian food. Jordan Fine, there he is. I remember you now. Gordy. You're a sports person. Rocky and Vanessa Gordon right next to each other as usual. The inseparable duo. Where is Azerbaijan? She's not come yet. Who's NK? Who's NK who just showed up? Let me see your face. Let me see your face. Okay. Nishi Kenderwall. Oh, okay. Yes. I've read your name many times. It's funny. What a weird year this is. I feel like I know you all by your names, but not your faces. Bemalak's been around forever. I've had you since Bio 101, I think.
Yep, that's true.
Are you back here or are you in Ethiopia?
No, I'm in Plattsburgh.
You poor bastard. I'm sorry.
I know.
Zach. Hey, Zach. What's happening, Zach?
Hey, Professor.
You don't have to call me professor. Just call me doctor.
Doctor, got it.
I have too much fun. They shouldn't be paying me for this at all. All right. Well, as long as we've got most of us here, if not all of us, let's go over the syllabus. I don't know if you had a chance to see it because I had trouble with the Moodle site. Just got it up about a half hour ago. So I will screen share my syllabus. Okay. Okay, so developmental biology, that's our class. And my office is 325 Hudson, although at this point I plan to be there about once this semester. If things go the way I hope they do, I'll only be there once. So anything you need, either email or call me. This is my actual cell phone number here. Another thing about this cell phone number is if it's like, 5 after 1 and I haven't shown up yet, I'd really appreciate it if someone called me and got my head back in the game. Sometimes I just forget. Sucks getting old. Okay, office hours. We're supposed to post 3 3 hours a week, but I mean, if you need me, just send me an email. We'll do a Zoom. It's not a problem. We can do it anytime. Here's your class Zoom link, which is also in Moodle. So there's that. So what are we going to do? We're going to talk about development. So in chapter 3, we talk about just kind of the morphology changes, the broad structural changes that happen during development, going from, fertilized egg all the way to an adult or child or whatever. And then we get into, starting with chapter 4, we get into the molecular stuff, which is my favorite part. And that's what a lot of this class is going to be about, is like, first you see how things move, and then you find out why they're moving. And it typically has to do with hormones being released by one tissue, received by a different tissue, and that different tissue responds to it by either moving or changing cell type. So that's most of what this class is about. And I think it's really cool. I really enjoy it. So anyway, chapter 4, we're going to get into Xenopus molecular development, and then we'll have an exam February 22nd. Now, again, I don't know. I think February 22nd was based on the normal schedule, but we got pushed back a week. I don't know if I changed that or not. So we may end up pushing that back. We'll see how long it takes to get through the material that I planned for that exam. So that may end up being moved. They all may end up being moved down 1 week. We'll see. Okay, so then exam 2 is chicken mouse development, and then morphogenesis, which is change in form in the early embryo. Exam 3 then is cell differentiation and stem cells and organogenesis. Actually, I think last year when I did it, I didn't get to all this material. So we'll see how the semester goes. I may not cover all of this. I'd really like to get to evolution and development, because that's really neat. It's like once the pattern is laid out, how does it change to give you a different pattern in a different organism? And that's really important evolutionarily, because as you'll see, everything is so tightly controlled that if anything goes wrong, the embryo just dies. So how is it that it can actually change and then give you a different organism after the mutations? So anyway, I'm not sure how much of this we're going to get through. We'll do as much as we can. Okay. And this is my disclaimer, which I always throw in there so I can't get sued. The schedule may change. I may just fail you all just because I'm not in a good mood this semester. That happens. It's my prerogative. Course objectives. I really hate these course objective things. It's like, does anybody read this? Does anybody care? But anyway, they want me to put it on the syllabus, so there it is. You can read through that if you want. Attendance, I would advise coming to these lectures. I will be recording them and putting them up on YouTube so you can look at them later. But A or B, make sure you're looking at them, because the textbook is pretty complicated. It took me a while to decipher it and then translate it into English. So you really want to come to the lectures so you can tell what's going on, or at least how I understand it. Hell, I might be getting it wrong, and and you have to learn it wrong to get it right on the exam. So no, I'm just kidding. You should definitely be coming to class. Although I'm not taking attendance, so if you can't make it or whatever, you'll get the link on Moodle. Now this is from an old schedule. This doesn't really count. Your exam, you can be out of town for the exam because the exams are all going to be online, so that's not that important. It's important though to take that exam when it's due. If you can't make it at that time, let me know. We'll work out a different exam schedule for you. So the class then is basically just going to be 4 exams. And they're not cumulative. They're kind of cumulative in the sense that the things you learn early are important for what you're going to learn later. So in that sense, they're cumulative, but I'm not going to go back to previous chapters and pick out specific topics and quiz you on them. So it's basically just 4 exams going through. And there's your grading scale. What else do we got here? Electronics use policy. This doesn't really count either because you're not in class. So I'm not worried about it. And you'll turn your video off soon. I won't know if you're using it anyway. blah, 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 blah. This is other stuff they tell me to add. So, okay. So basically in terms of syllabus, what's important is we're probably going to cover most, if not all of this. There's your Zoom link and my phone number. Don't be afraid to use that if you need me. Okay, any questions about the syllabus? No questions? Okay. Let's do a new share. Okay, so here is my PowerPoint. This is the textbook. Let me see if I can make it bigger. Okay, so this is the textbook. There's a link in Moodle that tells you how to get to it. It's online. I guess you can get a hard copy. The hard copy is probably $250. The online version is like 50 or 60. So I would definitely suggest going that way. Should you buy it? Everyone says, do I really need the book? I don't know. I mean, everything I talk about is going to be on 100 different websites. You do Google search, you'll find it. The information is the same whether you get it from this textbook or from some website somewhere. So if you're the kind of person, which I hope you are, I hope you all are, the kind of people that when you need something, you know how to go out, use Google and find it and figure it out that way. If you're that kind of a person, you'll be fine. If you want to read along the book with us as we go, I go straight from the book. So it might help for you to have the book too. So, but that's up to you. Let's take a quick look at the Moodle site in case I forget. Oh yeah, there's something I wanted to show you about the Moodle site too. Okay, so if you haven't gone to your Moodle site, which you probably haven't because it wasn't up until very recently, let me walk you through this a little bit and you can see how it's set up. So basically it's set up based on exams. And I think now that I remember, last year when I did it, chapter 3 was exam 1. So we only do chapter 3 for exam 1. There's a lot of stuff in each of these chapters. So what I have here is my PowerPoint, which I may change a little bit and I'll delete the old one and upload the new one as I change it. So there's that, that's important. Here's a Word file with all the vocabulary from the chapter in it. That's really important. Before, when you're studying for the exam or when you're just studying, you should be studying every day we have a class. And when you do, you should pull up that vocab and make sure you know which words are important. There are a lot of important new vocab words in this class. So that kind of gathers them together. And on the exam, I do a lot of fill in the blank kind of stuff. So it does help to know the vocab. And now after that, we've got all these links to animations. And I think animations are really, really important in this class because so much of it is visual. I can tell you about the pathway of a hormone messenger interaction and write it down on paper. It makes sense. But to see gastrulation, it's really hard to explain gastrulation unless you can see it in 3 dimensions and see what's going on. So these videos are really, really important. And if you come across some videos you think are better than mine, send them to me and I'll put them up on the Moodle page. So there's exam 1 is chapter 3, exam 2 is chapter 4, exam 3 is chapter 5, exam 4 is more of chapter 5. 5 was a really big, complicated chapter. So I spent a lot of time on it last year. And then chapter 7. Okay, so that's what we're going to be going over. So what you'll see up here in your Moodle page is, well, let me show you later, is your PowerPoint, your the notes from the lecture that I'll be posting, vocabulary, and the Zoom links, Zoom recordings that I'll put up here too, and other stuff that I throw in here just to make your day more pleasant. Okay. Questions about the Moodle site? Nothing? Okay. Stop that share. And now we're going to actually learn something today. You ready? Who's ready to learn, people? Oh, you all are. I know. You're all good kids. Okay, I'm just bringing my notes up so I don't forget anything as we're going through here. Come on and zoom, zoom, zoom. Every time I come up on Zoom, I think of this really bad PBS show from like, oh God, mid 70s called Zoom. It had this, it was kind of like, it was supposed to be for kids that are past Sesame Street. So it was like the next level of PBS schlock. And they had a theme song. And every time I pull up Zoom, I think of that theme song. It drives me nuts. Don't get old, people. Okay. So development, what are we talking about? So what we talk about here is all vertebrate development. Okay. I think chapter 2 is about Drosophila development. I'm not that interested in it. It's very different from vertebrate. So I decided not to cover it. I may cover it someday, but not right now. Mainly what we're talking about, we're only talking about vertebrates. So the vertebrates we'll be talking about will be the model organisms from different phyla, I guess. That's not my science either. But you've got amphibians, fish, the zebrafish, chick, mouse, and we'll do some human too. Not a lot of human, but enough to show you the difference between human and mouse. So we're not going to do a lot of zebrafish. It's not really covered that much in the book. We'll do some at the beginning just to show you how things like gastrulation are different in different organisms. Mainly, we're going to be focusing on xenopus, chick, and mouse. And it's really important that if you don't know which organism you're on, let me know, because things are very different between chick and mouse, and sometimes it gets confusing. So stop me, ask a question. Blah, blah, blah. So what is the vertebrate body plan? So, based on the first amphibians that crawled out of the water, the vertebrate body plan is a brain case, which could be bony for most of us, could be cartilaginous for some fish. And then you've got an axial skeleton, which is your for us, our arms, front legs for walking animals that walk on all fours. And then you've got your pelvic girdle. So you've got your shoulder girdle and your pelvic girdle. And again, based on that first amphibian that crawled out of the water. Hang on a second. Oh, go away. I can't. Oh, all right. Never mind. You can't see what I'm doing, but it's my PowerPoint is being annoying. Okay, so what this tells us then is we've got 3 different axes of symmetry, which I believe. Oh, before we go into that, here's our axial skeleton. So the axial skeleton is the central nervous system, brain case, spinal cord, plus all the bones that encase it, and the rib cage. That's your axial skeleton. And then the appendicular skeleton are your appendages. That's where the name appendicular comes from, and that's your shoulder girdles and your pelvic girdles. And axes of symmetry. So this is really important too. Early animals are mostly round. They've got one axis of symmetry. Some animals don't have any axis of symmetry, like sponges and corals just kind of grow off in different angles and stuff. There's no symmetry at all. Evolutionarily later organisms tend to have one axis of symmetry. So this has got a top and a bottom, but anywhere you look into it, it should look approximately the same. So radial symmetry is basically one axis of symmetry. And we all start out with just the one axis. When the sperm hits the egg, you've got one axis of symmetry. Here is our 3 axes of symmetry. You've got your dorsal versus ventral, so your back and your front. You've got your anterior versus your posterior end. So that's the head versus the tail. And then you've got right and left that are coming in and out of the screen here. Those are 3 axes. And those 3 axes are going to be set up by gastrulation, which is a really important event that we'll talk a lot about in this class. So your 3 axes of symmetry then, anterior, posterior. Sometimes they'll be called cranial versus caudal. I usually call them just anterior, posterior. Dorsal ventral, which is your back versus your front, and then left and right. Body planes has to do with, as you take a slice through that body to look at different structures, how is that slice made? So sagittal is this slice coming right down through the head and giving you that, that aspect. Coronal is coming this way and giving you that aspect. And then transverse is this way. So another thing about this class, you need to get good at visualizing 3D from a 2D picture. So you'll get used to that after a while. Okay. Life cycles, vertebrate life cycles. Just moving stuff around here. Okay, so here we're talking about xenopus, zebrafish, chick, mouse, and human. And here you can kind of compare the size of the egg. I think this line is like 1 millimeter or maybe 1/10 of a millimeter. Yeah, 1/10 of a millimeter. So xenopus egg is pretty big. Zebrafish is quite a bit smaller. Chicken egg is huge. We've even had to make that thing smaller. Mouse, your mammalian eggs are really, really tiny. And you will go from that single cell stage to a blastula, which is a hollow body of cells in the xenopus. This hemispherical cap in zebrafish, a hollow disc in the chick, and this cup shape in mouse. And then the human actually looks more like the chick. It's kind of a cup, a hollow disc. And then once you get post that stage, you can see they're all a little bit different at this stage. Then you get the phylotypic stage. So they're all in the same phylum, which is, I don't know what the hell the phylum is. Oh, wait, I do. Chordata. It's the phylum Chordata. Watch me come up with that when I needed it. So at this stage, they look a lot more similar. Okay, you've got a head, spine, and tail. Okay, and then after the phylotypic stage, they then develop differently. So that transition then from the blastula to the phylotypic stage is really important because, again, anything changes along that way. It's such a complicated process. If anything changes, then the organism or the individual just aborts, right? The embryo doesn't make it and it dies. That's a lot of what miscarriage is. Early on, divisions go wrong and the organism's non-viable and then it aborts. Okay, what am I doing here and why? Let me check my... I'm not sure why this one was here. Hang on 1 second. Don't move. Oh, that's neurulation. Why am I doing this? Okay. Well, anyway, I'm not sure why I have these slides here, but let's talk about them anyway. This is all neurulation. Yeah, we'll talk about this later today, so let's not worry about this now. Let's start in with xenopus development. I used to work with xenopus development in my second postdoc back in the early 90s. And it was really, really kind of fun model organism to work with because it's all, outside the body. It's really hard to work with mouse embryos because they're all inside the body. Chick embryos are inside the egg, which makes it a little more complicated, but frog embryos are really easy to work with. So basically what you do is you take the adult female frog, you inject it with HCG, human chorionic gonadotropin in the evening. You go home, you have your night, come back in the morning, and she's all ready to go. You carefully grab the female and you stroke her sides, and the eggs come flying out the back. And that's what the male does. Male's smaller than the female, latches onto her and squeezes her, and the eggs come out. So normally he's sitting there dumping sperm on him as the eggs are coming out. So it's an external fertilization, which again makes it a great experimental model because we can do it ourselves. I would do that with the female, squirt the eggs out into a dish. Now you've got a whole bunch of eggs ready to be fertilized. And then, unfortunately for the male aspect of the project, you have to euthanize the male, kill him, pull the testes out, crush the testes up in a little tube, and the sperm come out. And you take a little dip of sperm, you drop it on the eggs. And the eggs are kind of all different orientations. One top is brown, the top is brown, the bottom is yellow. But when you add that, before you add the sperm, when they're just eggs, they're kind of twisted in all different directions. Once they get fertilized, boom, they all turn. And so they end up with the animal pole on top and the vegetal pole on the bottom. So so if you're going to be if you come back in life as a xenopus, come back as a female xenopus, because the males don't make it for very long. So anyway, here's our egg. Here's our xenopus egg. Better picture of it. There it is. You've got this brown portion here, which is the animal pole, and the yellow portion, which is the vegetal pole. And the bottom is yellow because it's full of fat. It's the yolk. So this is the yolk of the cell, although there are important mRNAs and things here. It's not just fat. This will be, these will become cells as well eventually. And the animal portion is here. And if you shine light through it, you can see like a silvery halo around the whole thing. And that's a really thin membrane called the vitelline membrane. Now, when you add your sperm to these eggs, they will always come into the animal side. Okay, they won't enter through they may try, but they won't get in through the vegetal end. They always will come in the animal pole. And the point where they come in is really important. We'll talk about that in the next chapter, how that will determine the axes of the animal. Okay, so once that sperm enters, the egg is not yet ready to be fertilized. Sort of, it's gone through one round of meiosis. Okay, so it's gone through replication of the DNA. First round of meiosis, you get one polar body, which is not fertilizable, and then the egg. Now this egg has to go through a second round of fertilization, but it's like frozen at this spot until it gets fertilized. When it gets fertilized, the first thing that happens is you go through a second meiotic division, you get a second polar body, which ends up just getting lost. And now you've got a nuclei here that's ready to fuse with the sperm nuclei. So right after that sperm enters, boom, the cell goes through its second meiotic division. Now it's got a pronucleus for the female and a pronucleus for the male that fuse. And now you're rocking and rolling. That's your zygote. Okay, so here you can see the polar bodies up on top. Here's your sperm entry point. Okay, so once that happens, the cell starts to divide. So your one cell here goes through mitosis and it divides into two cells. So in this like a vertical cleavage point. Now at this point, both of these cells are identical, right? They've got the same nucleus that was replicated once. So you could take these cells and separate them, you get two identical frogs out of it. Here again, you cut in another vertical direction. Now you've got four cells that are all identical. Split them apart, let them go. You'll get four clones of the same frog. Who wants four of these stupid frogs though? Sorry. So you've got four cells now and they're all identical because they've gone across that vertical plane. And we'll explain in the next chapter why that keeps them identical. And this changes it. Once you get this horizontal cleavage, Now you've got brown cells on top and yellow cells on the bottom. And as we'll see later, you've got different messenger RNAs that are attached to the cytoskeleton in the top and bottom, and that'll make these cells different. These cells on top will be ectoderm, and the cells on the bottom will be endoderm. So those are our first two, I guess you call them proto-tissues, the germline tissues, the ectoderm and the endoderm. The ectoderm will go on to develop into nerve and skin, basically. And the endoderm will go on to become gut. So here you are, 1, 2, 3 divisions. You've got 8 cells, and that will continue to divide and divide. At this stage, we kind of call it a morula. I think this is a morula as well, which is just a whole bunch of cells stuck together. I think the morula stage is really more important in mammalians than frogs, but we can call that a morula. And then once it goes past morula, it gets to this blastula stage. In the blastula stage, you've got a hollow cavity in the middle. And by hollow, I mean it's filled with fluid, not air. So basically, the cells have gone through many, many rounds of division without growing at all. Like normally in an adult, a cell will divide into 2, and then each of those 2 will grow up bigger, and then they'll grow even bigger before they divide into 2 more. Here, they're just dividing, basically just the nucleus is replicating and dividing. So you get many, many small cells. So especially the ectodermal cells on top, they'll be smaller than the endodermal cells. So you've got ectodermal cells on top and your endodermal cells on the bottom. And by the time you hit this blastula stage, you've got a third tissue that's been induced. And this is at the marginal zone. So right around the center of this hollow sphere, you have a third tissue, which is mesoderm. Mesoderm will go on to become muscle, blood, bone, and basically everything else that we didn't get with ectoderm and endoderm. So that's your third tissue. So right now, this is the last stage where you're going to have a single axis of symmetry. You've got, just like the sea anemone or whatever, you've got one axis of symmetry. Anywhere you look at it from the other directions, it's exactly the same, one axis of symmetry. Now we're going to gastrulate and get our 3 axes. This is kind of the beginning of gastrulation. So gastrulation involves here at one side of the embryo, cells start to invaginate here, or involute. So this is early blastula. Check my notes to make sure I haven't forgotten something. Okay. Okay, so this is called the blastopore lip, okay? We're in the blastula stage, and this is going to form a pore into that blastocele. So this is your blastopore lip, and this lip part here, the dorsal lip, is your Speeman organizer. This will be really, really important in the next chapter. This is going to be giving off hormones that are going to cause the dorsal axis to occur, cause it to happen. So looking at it just kind of morphologically though, we see these cells are twisting in and then going up. So cells keep coming in. Let's see, they're coming in from here, and from here, they're kind of converging on this spot, and they go up and in and around the top of the embryo like that. So here you can see them starting to go in, and we've got mesoderm and endoderm crawling up like this. You can see a layer of red and a layer of yellow, and it's crawling in. Let me see if I've got a bigger one. I think I do. Okay, so here's your blastopore lip forming, Speeman organizer. Cells are coming in like this. So they're coming down the mesoderm like that, and the endoderm's coming up. So you've got this dual layer of red and yellow that's crawling up this way. And you can see there's a gap between these 2 layers of yellow cells here, 2 layers of endoderm. And that drags itself up this way. Once it gets all the way to the other end, that's your gastrula. Now that hollow gap there between the layers of endoderm will be the inside of the intestine. So remember, I said just a few minutes ago that endoderm is the gut. Well, here you can see the primitive gut forming. You've got endodermal cells surrounding this cavity. Here's going to be the anal exit, and the embryo will eventually give itself a mouth over here. And so this is the dorsal now. Ventral is down here. You've got anterior here, posterior here, and then left and right in and out of the screen. And here at the end, you've got the yolk plug, back by where the blastopore lip was. And here's the blastocele, which will get pushed out that way and it'll get lost. So the blastocele doesn't survive much past gastrulation. Okay. Okay, so another thing that's going on here at the same time that these inside movements are happening is on the outside, the ectodermal cells are going to cover the whole, I don't see it here. We'll see it in the video in a minute, but basically these ectodermal cells are dividing and dividing and dividing, right? They divide really fast. So throughout this gastrulation process, these cells are pushing down around, you know, in 3 dimensions in every way. They're pushing down around this way. You know, here it is. Now you can see it. These blue cells are pushing down here, and they're pushing down here, and these surface blue cells will converge on that that yolk plug there. So now you've got ectodermal cells surrounding the whole embryo, which again makes sense because ectoderm is going to be skin. You want skin on every external surface. So that process, which is called epiboly, is those ectodermal cells dividing, and then pushing down around the whole embryo until the whole embryo is covered in that ectoderm. Okay. By the way, if you have any questions, do we have a chat? We've got something in the chat. Is there a way to post the Zoom link on Moodle? Oh, it is up there. This must have been from earlier. Hopefully that person found the Zoom link. Okay, if you have a question, go ahead and post in that chat. I can see that. Okay, where the hell am I now? All right, so now you've got your gastrula. And another thing that's going to happen here is that it's going to start to extend and elongate. So it's not going to be a ball anymore. It's going to start to stretch out. Now, the next step after gastrulation is nervulation, the formation of the nervous system, really central nervous system, and the rest of it will follow from there. We've got a chart. Vanessa. Oh, when am I uploading the lecture on YouTube? It should be there now. Normally I'll have it up there before the class starts. I think I forgot to do it today. Let's see. Yeah, today's notes are not up there. They will be, oh, I can do it right now. That's the kind of guy I am. Let's do it right now. Coming at you live from Burlington, Vermont. Okay, so again, I apologize for not having them up already, but now they are in your Moodle site. They're the last link. Well, let me move it up where it belongs. Nervulation. Little handle there. Get a handle on it. Get a grip, Slish. Okay. So it's up there in your Moodle now. Okay, so nervulation. I know someone was asking me about this. So what happens now is once you've got epiboles happen, so you've got ectoderm all over the top, you've got a layer of mesoderm underneath that ectoderm, and now you're going to have basically talk between the mesoderm and the ectoderm. So hormones are going to be passed back and forth, and now what is directly under the midline of the mesoderm, right underneath the midline, will cause this ectoderm to become neuroectoderm. Remember, ectoderm can become nerve or muscle. this is going to change the stuff in the very center into neuroectoderm, which means the ectoderm on the sides then is epidermal ectoderm. Okay, so we're starting to see differentiation from these original germ layers into more specific tissues at this point. That's differentiation, which is what this class is all about. So because you've got this mesoderm here, it's releasing substances, which is causing the ectoderm to become neural in character here. Okay, so that mesoderm then becomes what's called a notochord. So the notochord it's a bunch of cells, mesodermal cells, that will direct the development of the nervous system. And eventually it will become part of the spine. I think technically it's mainly the discs between your spinal cord. And then on either side of the notochord, you're going to get something called somites. And those somites will become the bones of the spinal cord, as well as all the axial structures, the ribs, the muscles, and then the arms and everything else. So at this stage, though, in the middle of neurulation, you've got a notochord here, somites at either side. The ectoderm has become neuroectoderm, and now you've got epidermal ectoderm on either side of that plate. And now it starts to fold up. So we call these bumps neural folds as that starts to turn inward. And those neural folds will come up from one end to the other until they come together and fuse at the top. Let's see if I have a better picture of that. Okay, so here's kind of like early and then late neurula. Early neurula, you've got your notochord here, and this becomes neural plate, and then epidermal ectoderm on either side. This pushes up. The neural plate closes up and forms a tube. We call it the neural tube now. And the epidermal ectoderm covers over the whole neural tube. So that's this thing coming up, sealing, and now you've got your brain and spinal cord encased in skin, and the notochord is underneath it, plus your somites at either side. Here's a really nice version just looking at the ectoderm. Here's neural plate, and these cells at the corner start to bend, and that pulls the thing up together. It fuses at the top, and then you have your neural tube with ectoderm across the top of it. So that's neurulation. Any questions? Anyone want to chat a question? Okay, so another important part of this, right here at the end of neurulation, you can see this little bit here. Let's blow it up. So here's your neural tube, epidermis across the top, notochord underneath, and here are your somites on either side of it. But over here, when we're looking at the nervous part, the tube is closed up and you've got skin over the top, but we've got extra cells here from the top of the neural tube. So you can see these cells here where they're going to fuse. After the epidermis fuses, you get these cells called neural crest cells. Now, neural crest cells are mesenchyme cells. Mesenchymal cells it's a general term that means cells that move around and go somewhere else. Okay, so these guys here, the skin are all attached to each other. They're not going anywhere. The neural tube cells are all attached to each other, but these cells let go. And once they let go, they start to crawl. And as they crawl away individually, they will then form other structures. They'll form the peripheral nervous system, glial cells to help with the neural system. Melanocytes come from neural crest. So all the skin, all the color in your skin comes from these neural crest cells that then crawl out and get into your skin. A bunch of other things come from these neural crest cells. Okay, so that's your neurulation. Neurulation. Variolation. See if I forgot anything. Okay, so once you get to this late stage, there's another couple of structures you need to know about. So here at this stage, we're going to blow this up and give you more things to memorize. Okay, there, okay. So we're at this stage, okay, the late neurulation or the end of neurulation. Hang on, I've got a chat. What is the function of the somites? Good question. So the somites we'll talk a lot about later. Basically, right now, they're going to be mesoderm. They are mesoderm that will form structures of the skeleton. And actually, I'm going to talk about that in a second here with this stuff. But basically, you know, notochord and neural tube is all axial, and this is going to form everything else. The ribs, the arms, the legs, and everything else are going to come from the somites. Everything that's mesoderm. Okay, so look at this bad boy here, okay? You still got, here's your gut. All that yolk is right around the gut, so you're gonna eat that yolk up. Skin all the way around, neural tube. Now I'm gonna blow up just this one part, and we'll talk about what those somites become. Okay, so you've got notochord in the middle, here's a somite, and here's a somite. So that's gonna become, actually, I think I was wrong a minute ago. Somites aren't going to become all of the axial skeleton. They're going to become the spinal cord, okay? Not the cord, the spinal column, the bones of the spinal column and the muscles along the spine. That's what the somites are going to become. But you've got other mesoderm here that's going to become that stuff I just said. So these are, let's see, check it before I make a mistake. Yeah, okay. So now you've got mesoderm over here that's divided into two different types of mesoderm. You've got your somatopleural mesoderm, which is on the outside, and that somatopleural mesoderm will become, oops, that will become the body wall and the limbs, okay? So this will become, you know, your ribs, all the muscles along your ribs, and your arms and your legs and things. And then you've got a space in the middle, the coelom. And the coelom then will be like the body cavity in between organs and things. And then underneath that is this splanchnopleural mesoderm. I hate that word. I can never pronounce it right. Splanchnopleural mesoderm. And that splanchnopleural mesoderm, you can see it's more central than the somatopleural mesoderm. So that splanchno will then become, get this wrong. Splanchnopleural will become internal organs, heart, blood vessels, mesoderm that's on the foregut, things like that. So in terms of the mesoderm at this neuro stage, neurula stage, you've got notochord and somites, which will become part of the spinal column. You've got neural tube, which will be the spinal cord. And then you've got other mesoderm here, which is going to become the outside of the body, the muscle and bone of the rib cage and the body structure. And then the inside, which would be the inside mesoderm things, heart, blood vessels, blood, things like that. And of course, your endoderm is down here. It's still just going to be gut. I believe also, I just looked this up yesterday, I think the urogenital tract is also mesoderm. So that's another thing that comes out of that mesoderm. Okay, so that is neurulation. And after neurulation, you get to the tail bud stage, called that because it's got a tail bud. And at this stage, you can really see the segmentation. And those are separate somites. So each one of these is a somite layer. And that segmentation is really important. And we'll, towards the end of the class, we'll see how that comes about. It has to do with Hox genes, and it's really interesting. And then the brain at this stage is also separated into different things: forebrain, midbrain, hindbrain, eye forming, adhesive organ to stick to things. So there we go. Okay, that's Xenopus. That's kind of the large morphological explanation of Xenopus development. Okay, any questions or comments? Oh, Azerbaijan has showed up. Yay, Isun. Hello. Any questions or comments about this? I definitely think you should read it over tonight, look it over. And again, you can get this anywhere. You can go to Wikipedia, you can just do Google searches on it. There's so many scientists out there who can't wait to tell you about gastrulation. So if you don't want to buy the book, do that. But the book will take you through it for $50. So it's all up to you. So that's all I have. Any questions or comments?
Should I wait after class or you'd send a new Zoom?
We'll just talk for a couple seconds after class. So I'll just keep the same link, okay?
Okay, sure.
Okay, any other questions? Okay, so you can all leave now. I have to talk to Tyler for a couple seconds here and then we'll be good. See you again on Wednesday.
Thank you.
I'll be missing you till then.
Thank you.
When are you going to upload the actual, like this lecture video to YouTube?
As soon as we end, it'll process and then I'll put it up on YouTube and then I'll put the link on, and definitely today, but not for at least an hour or two. It all needs to function.
I guess no rush. I just wanted to know because during lecture, some of the things, when I was taking notes, I missed some of the points and some of the information.
Okay. Maybe in the future, I'll make sure I get my notes up before class, and then, it'll be easier to follow along. Okay?
Okay.
Michelle, are you just coming? Michelle.
Hi.
Did you just show up?
No, I don't know what happened. I got kicked out at like 46 or 7, like right at the end for some reason.
Okay, well, I can program it to kick out people I don't like, so maybe that's what happened. All right, we're all set though. I just have to talk to Tyler for a couple seconds and we're all done.
Oh, okay.
Okay?
Yep, all right. I'll see you next class.
See you later.
Bye.
Okay, so Tyler, this independent study we're doing? Tyler?
Yeah.
Evans, this is just for Tyler.
Yeah.
Okay, so Tyler, this independent study, I'm still working on the code. I'm stuck on one little point. I hope to get it this afternoon, and then it should go a lot easier. So as soon as I get like the first general game put together, then we can start working on it. You can start putting information in, okay?
Okay, sure.
I hope to get it done by the end of the week, but you never know with coding. Sometimes it goes easy, sometimes it goes hard.
Yeah, true.
Okay?
Yeah, no problem.
Okay, we'll see you later.
Yeah.
Hi.
Hi, you want to talk about your letter of recommendation, right?
Yes, please.
I'll reply to your email, okay?
Okay, sure.
I can write it for you now because intro to the health professions the A doesn't mean much in that. Everybody gets an A, but the other one's important. So I'll write you a letter based on that one, okay?
Okay, sure.
Okay. All right. See you later.
All right.